So good afternoon, grade 8 STE students natin. So ayan, so we still have almost 7 minutes before we officially start our lesson for this afternoon. So kaninang alauna, nagconduct po ng online class for my grade 8 students regarding particle nature of matter. So for this afternoon naman, starting from 2:45 onwards, probably hanggang 4:30, we will be having our discussion for part 2 naman ng nucleic acids natin. So while we are waiting for those students who will be attending our class, let me check my audio kung okay yung audio natin. And mukhang okay naman yung audio at visuals po natin. After this class, ipapasa ko na po isasend ko na po sa inyo yung file o yung PDF file ng ating PowerPoint so that you can review your discussions before you answer the pre-test, before you answer activity 1 and quiz number 1. By the way, after this one, pwede nyo na rin sagutan po ang pre-test natin. Yan. So let me show some of the students who are currently now on our online class. Yan. So thank you for this one. I know na galing kayo sa online class din ng Science 8 niyo with Sir Edmond, but do not worry. 2:45 pa naman tayo mag-start. You still have 6 more minutes before we officially start our class this afternoon. Ayan, so I will flash the names of who are currently attending our class this afternoon. Ayan, thank you. Magkakaroon po ng kaunting changes do sa groupings ng performance task natin dahil nga po yung iba may mga announcement po may mga updates po regarding sa face-to-face classes para sa ating subject na biotechnology. Kaya i-update ko na lang po kayo sa ating mga respective GCs. But for the meantime, you may attend this online class ng ating subject. Ayan. Thank you again for those who are currently attending our online class this afternoon. Ayan. Thank you. Inom lang ako sandali bago tayo magsimula ng klase. We still have 5 more minutes. I hope that you have, I just hope na na-review nyo yung discussion natin kahapon because that will be very essential sa part 2 ng ating lessons hapon na ito. Meron po tayong isang lesson na hindi na po isasali sa online class. You just need to watch the PRISM video regarding mutations. So you just go over sa mga links na shinare ko po sa PDF file and just watch that mutation topic. Madali lang naman yung topic na yun. So just watch that one. Kasi kasama siya sa first set of activities natin. So I will just again post. Yan. Thank you again for those who are currently attending our online class this afternoon. You may please invite your classmates na and schoolmates, especially grade 10, grade 11, and grade 12 to attend our class this afternoon kasi mapapakinabangan nila yung lesson na i-discuss natin sa oras na ito. So yan, thank you. I just hope that na bago kayo umattend ng ating class ngayon ay nakainom na kayo ng tubig dahil alam kong galing kayo sa isa pang online class sa inyong Science 8. We still have 4 minutes. Exactly 2:43, I will now be starting sharing the PowerPoint and we will be starting our class this afternoon. Yan. So, alam ko medyo naging information overload yung naging discussion natin kahapon. So, magkakaroon muna tayo ng review discussion before we proceed to our topic for this afternoon. Sa mga may agam-agam dahil hindi sila napabilang sa face-to-face o dahil napagdesisyon na ng kanilang magulang na hindi sila attend hindi kayo sasali sa face-to-face, wag po tayo mag-alala because the quality ng pagtuturo na ginagawa po namin mga teachers sa online class will be the same sa face-to-face. So kung naman yung ginagawa namin sa face-to-face, ganun din po yung quality na ibibigay namin sa mga online class at saka sa mga modular students natin. So don't worry about that one too much. Ang pinagkaiba lang naman is paano namin i-share sa inyo yung mga concepts na gusto naming ibahagi. So, yun lang naman ang nagkaiba doon. Sa inyo, online class, yung isa sa face-to-face, yung isa sa modular. So there's nothing wrong, there's nothing to worry about at once. So kung medyo magkakaroon tayo ng problema sa internet connection, just bear with me kasi medyo lumalakas ang ulan sa area namin, and I think sa inyong lugar din naman. But nevertheless, pwede nyo naman panoorin itong video lesson natin once we are done with our lesson. So just give me a minute. I will now be starting sharing my screen. Oops, sorry. I just hope that my audio and visuals are now okay na sa inyo, sa part ninyo. Because right now, naka-flash na sa aking screen. Ayan, just give me 2 minutes to finally flash completely the screen ng aking PowerPoint. Ayan, I think okay na. So again, good afternoon, grade 8 ST students from Darwin and Mendel. So for this afternoon, we are going to have now nucleic acid part 2. So kahapon, na-discuss na po natin yung part 1 natin, which comprises the experiments that led to the discovery of the genetic material, as well as the role of nucleic acid and some parts of the structure of the DNA. For this afternoon, we will start with that one, structure of nucleic acid. Then we will end up with chromosome. Again, this is Mr. Marin Marinas, and I will be your teacher facilitator for this afternoon. So we will start with the structure of nucleic acid. So magkakaroon tayo ng review muna before we proceed to our main topic for this afternoon. So yan, meron po tayo ditong diagram ng nucleotide. I just hope that you still remember what is a nucleotide. So hindi ko sasabihin kung ano yung components niya. So yan, let me just, yan. So let's have number 1. Using the comment section, please type there anong tawag sa number 1 na ito. Ano kayang structure, ano kayang molecule itong naka-attach or rather naka-label as number 1 here. Please again use the comment section to say to state your answer. What do we call this number 1? Ano kayang tawag sa number 1 na ito? Again, please use the yan. Isaiah answered phosphorus group. Is that the term that we used yesterday? I-check natin ha, i-review natin. Ano kaya yung term na ginamit natin in this day? Tama po kayo, it's a group. It's a functional group. Pero ano kaya yung tamang term in this case? Richard answered phosphate group. Keisha also answered phosphate group. Tama rin naman yung phosphorus group because mainly it's composed of phosphorus. But the correct term is phosphate group. What about number 2? What do we call this number 2? What do we call structure number 2? Number 2 na tayo, ha? Ano kaya ang tawag sa number 2 na ito? Fast forward ko lang ng kaunti. Yan. What do we call number 2? Yan. Joshelle answered sugar. Pwede kayo maging specific if you want. Isaiah also answered sugar. Most of you are currently answering sugar. The answer for this one is sugar. In this case, this one is an example of deoxyribose. Deoxyribose siya kasi wala siyang oxygen. Wala ditong oxygen. May oxygen yan. That is ribose. Pero in this case, that is an example of deoxyribose. Next, number 3. What do we call number 3? Or molecule number 3? Ano kaya ang tawag sa number 3 na ito? So again, good afternoon sa mga humahabol sa ating session for this afternoon. So thank you for attending our class. So while waiting, so let's see what's the answer for number 3. What do you call number 3? Isaiah answered base, Richard nucleotide base or simply base. Marcus is very specific, nitrogenous base ang sagot niya. But most of you are currently answering base, which is also correct. Pero dapat na maging specific na tayo dito. This one should be called nitrogenous base. Nitrogenous in a sense kasi ito ay binubuo o meron siyang taglay-taglay na mga nitrogen atoms. Again, that is the 3 main components of a nucleotide. Phosphate group, sugar, and a nitrogenous base. In this case, that's adenine. Now, sa next part ng review natin, ang gagawin nyo lang po dyan is identify lang natin kung yan ba ay purines or pyrimidines. Kung maalala po natin, meron tayong dalawang uri ng bases or nitrogenous bases, the purines and the pyrimidines. So ang gagawin lang po natin dyan, i-identify lang natin dyan if that is purine or pyrimidine? Let's have cytosine. Cytosine, is it purine or pyrimidine? Again, use the comment section for your answer. Again, cytosine or letter C, is it purine or pyrimidine? Rian answered pyrimidine. What about the other students here? What do you think is the answer for this one? Saan natin ika-classify ang cytosine? Richard answered purine. Sige, okay lang po magkamali. Okay lang po iba-ibang sagot. I-check naman natin yan. Marcos, Jomel, Maika, Isaiah, Kisha also answered pyrimidine. For the number 1 here, the answer is pyrimidine. What about adenine? Adenine, is it purine or pyrimidine? Jane answered purine. What about the other students here? Joshelle answered pyrimidine. Sige, tingnan natin kung sinong tatama. Magkaiba kasi ang sagot nila. Jomel answered purine. Yan, purine. Rian, Marcus, Colleen, Jamil answered purine. The answer for this one is purine. What about thymine? Thymine, is it purine or pyrimidine? So Rian answered pyrimidine. Okay, Jomel, Marcus also answered pyrimidine. The answer for this one is, yan. Thank you for answering. Thank you for your responses. The answer here is pyrimidine. What about uracil? Uracil, is it purine or pyrimidine? Okay, what about uracil? Uracil is only found in RNA. Again, kung naalala niyo, pag sinabi nating RNA, it is a single-stranded polynucleotide. The answer is also pyrimidine. And lastly, we have guanine. What about guanine, which is G? Guanine, is it an example of purine or pyrimidine? The answer is purine. Now, before we proceed to our discussion ano nga ba ulit ang pinagkaiba ng purine at pyrimidine? So, para matandaan lang natin ito, pag sinabi natin purine, purine, it's PAG. It's purine, adenine, and guanine. Pag naman pyrimidine, it's PICAT. Cytosine, uracil, and thymine. Anong main na difference between purines and pyrimidines? Let me ask you sa comment section, what's the main difference between these two types of bases? Between purines and pyrimidines. Ano kaya pinagkaiba nila? Again, please use the comment section. What do you think is the main difference between purines and pyrimidines? It's very obvious in their structure. Ano kaya pinagkaiba? Both of them contains nitrogen atoms, but I'm asking you, what is the main difference between purines and pyrimidines? Anong pinagkaiba nila? Jane answered purines are larger, definitely. Kitang-kita naman natin. Rian answered single ring daw ang pyrimidine, which it is correct. Maika answered purines are double ring. That's correct. So tama po yung mga sagot po natin. So definitely, purines are larger compared to pyrimidines, while purines are double ringed, while pyrimidine is a single ringed nitrogenous basis. So thank you for your responses para sa ating first review. Let's have a quick rundown of what we have discussed yesterday. So sabi po natin, nucleic acid are macromolecules, meaning they're heavyweights. Mabibigat na mga molecules yan that exist as polymers called polynucleotides. And each polynucleotide is definitely composed of monomers called nucleotides, which again, na-review natin kanina, is again composed of phosphate group, pentose sugar, and a nitrogenous base. Again, this part is a review kasi na-discuss natin ito yesterday. So yan, nabanggit na po natin kanina that the three main components of a nucleotide is a pentose sugar, a phosphate group, and a nitrogenous base. Okay, so in this case, pag sinabi nating nucleoside, it is composed of only the base or the nitrogenous base and the sugar. Pag sinabi nating nucleotide, it is composed of sugar, the base, and the phosphate. Okay? So I just hope that you recall these terms na binabanggit natin sa oras na ito. I think nagkakaroon tayo ng problema sa visuals, parang lumabo. But I think don't worry, I can send this file naman mamaya, itong PDF file nito. So ayan, na-discuss natin kanina sa review yung difference between nitrogenous bases. These are nitrogen-containing molecules that act as bases. We can classify them into pyrimidines and purines, and the main difference is that pyrimidines are smaller or lighter. They are single-ringed nitrogenous bases, while purines are double-ringed, larger nitrogenous bases. Skip na natin itong mga to. Yan. Another difference between DNA and RNA is that aside from their number of strand, the sugar for DNA is called deoxyribose. Deoxy meaning without oxygen. Here, in carbon number 2. Wala siyang oxygen doon. Kasi sa ating ribose, meron siyang OH or yung tinatawag nating hydroxyl group. Hydroxyl group. OH is hydroxyl group. If you still remember our discussion during your first grading period. Now again yung mga carbon numbers here is numbered as 1, 2, 3, 4, and 5. 5 carbon sugar po, pareho ang deoxyribose and ribose. And please take note of these numbers because this one will become very handy kapag diniscuss na po natin ang ating other parts ng topic for this afternoon. Yan. I think this one is okay na. Let's move on. Yan. Dito na tayo mag-start. Sabi natin kanina that nucleic acids are macromolecules. If you still remember our discussion during your first grading period, pag sinabi nating macromolecule, it is a polymer. Isa siyang malaking molecule na binubuo ng mga monomers. In the sense of proteins, meron tayong amino acids, and each amino acid will be linked to each other forming a protein or polypeptide, as well as ating carbohydrates, wherein the monosaccharide units will be attached to another monosaccharide forming a polysaccharide, which is an example of a carbohydrate. Now, every time na magdudugtong daw ang isang nucleotide sa isa pang nucleotide, it is involving what we call dehydration reaction. Every time na nagkakaroon ng dehydration reaction, H2O will be produced. And as you can see here, linawin lang natin ito Ito muna tayo Carbon number 5, sabi natin kahapon, dito naka-attach si phosphate group. Liwanag tayo dyan Again, the phosphate group is attached to carbon number 5. Now, in a dehydration reaction, the other end of the phosphate group will be used to attach to another nucleotide. But in this case, it will not attach to carbon number 5. It will be attaching to carbon number 3. So, napansin niyo yung pattern, 5-3, 5-3, 5-3. Ganun din po sa other chain ng nucleic acid natin. So, dito pa lang, makikita na natin ang isang unique property ng strand natin. There is what we call directionality. Meron siyang 5' end at 3' end. In this case, this is your 3' end kasi open siya. In this case, we have 5' end. So maliwanag tayo sa part na yan. Due to this linkages or linking of one nucleotide to another nucleotide, there is what we call directionality sa ating nucleic acid. It's a 3', 5' direction. The bonding, yung pagdudugtong nito, will result in a repeating pattern. In what repeating pattern composed of sugar and phosphate units, which is now called sugar-phosphate backbone. As you can see here, ito yung sinasabi kong directionality. This is your 3' end or carbon number 3, carbon number 5, carbon number 3, 5, 3, 5, 3, 5. So meron tayong 3' end here, 3' end, and we have a 5' end here. Pasensya na, hindi ko na i-connect yung drawing tab ko. Next week, i-connect ko na ulit yung drawing tab. Nahirapan na mag-drawing. So, but nevertheless, in this single strand of nucleic acid, you can see here that one end is 3' and the other end is 5'. So dyan pala makikita na natin yung directionality ng ating nucleic acid. And this one will be very handy once na diniscuss natin later yung iba pang properties ng nucleic acid natin. By the way, this repeating pattern will form the sugar phosphate backbone. Sugar, phosphate, sugar, phosphate, sugar, phosphate. It's a repeating pattern. At nasa loob niya yung mga yung nitrogenous bases natin. Okay? Should be clear with you right now na yan, ha? Let's move on. Ayan na. Ito a little bit complicated na tong part na to. So I will go over here very slowly. The two pre-ends of the polymer are distinctly different from each other. Maliwanag yan sa diniscuss natin kanina. Bakit? Because one end is 5' and the other end is 3'. This goes along with the other side, 3' and a 5' end. And as you can see here, ano napapansin niyo sa arrangement nila? Let me ask you sa comment section. Can you tell me what did you observe? Kasi yung isang strand is 5' end, 3' end. What can you observe? Let me ask you sa comment section. Anong napapansin niyo po sa ating DNA? I just hope na maayos yung klaro yung ano natin. Medyo malabo yung ating connection o yung ating visuals, but I just hope that you can still see the diagrams here. But do not worry, kung nahihirapan kayo sa visuals, I can send you the file later. I will send you the file later. So kung nahihirapan po tayo, let me just continue na rin. Baka nahihirapan po tayo. Check na natin. Sabi daw ulit natin dito, the two free ends of the polymer are distinctly different from each other. One strand is running 5' to 5' going to 3'. The other strand is going the other way. The other strand is running from 3' to 5' end. So napansin nyo, magkasalungat yung kanilang direction. Though it shows directionality, mapapansin po natin na magkaiba yung direction na tinatahak ng ating dalawang strand ng ating DNA. One is running 5' to 3' and the other one is running 3' to 5'. Okay? So, one end has a phosphate attached to a 5' carbon and the other end as a hydroxyl group on a 3' carbon. Ito yung sinasabi ko po kanina. That's the technicalities ng ating description na binabanggit natin. We refer to this end as 5' end and the 3' end of our nucleic acid. I just hope that you're still with me at this point. Now, the two sugar phosphate backbones run in opposite 5' to 3' direction. They're always 5' to 3' direction. Ang problema lang is yung orientation nila. One end is running here, 5' to 3'. The other one is still 5' to 3', pero mapapansin nyo, sa ibang direction na siya. The opposite direction na siya. This orientation is what we call anti-parallel. Okay? And this one will be very significant once na diniscuss na po natin yung mga susunod na discussion natin sa lesson number 2. This orientation is what we call anti-parallel. Yes, they are parallel lines. Pag sinabi natin parallel lines sa math, di ba? These are lines that do not meet. Tama naman. The sugar phosphate backbones are just like lines. They're parallel lines, pero they run into opposite direction. Kaya sa tinatawag na anti-parallel orientation. That should be clear with you right now. Okay? Aside from that one, itong lesson kasi natin ngayon is more on more on structure. Yung physiology or rather the function will be discussed further next week. Yun yung medyo, what if nagparallel po ang direction nila or same ang direction? Hindi ko masabing impossible kasi pwede nilang gawin yan sa laboratory. In natural setting, it runs anti-parallel, okay? Because of the orientation of the molecule itself. Yung pag-prepare ng mga bases na yan sa gitna, ito yung technicalities bakit naging ganon. Yung mga pairings ng bases sa gitna na yan, yun ang isa sa mga naging dahilan kung bakit nag-swap yung nag-switch yung orientation ng isang strand. Di ba naalala nyo kahapon di ba pinagtatalunan lagi ni Watson and Crick ano nga bang klaseng orientation or anong klaseng structure meron sa DNA? So nahihirapan sila sa part na yun. So they tried several models and they found out na ganyan yung working structure ng ating DNA. Now, aside from that one, the linear order, yung pagkakasunod-sunod daw ng mga nitrogenous bases sa isang gene specifies the amino acid sequence, the primary structure of protein, which we will discuss next week, part 2. Medyo mahaba kasi ito itong transcription translation process ito. It's a very complicated process. Typically, 2 linggo namin dinidiscuss ito sa aming online, sa face-to-face classes. Okay? So, iniisa-isa talaga namin ito. Pero for the sake of convenience at para makuha nyo yung main concept dito, mas padadaliin natin ang kaunti ang discussion natin next week. So do not worry about this one. So ibig sabihin yung pagkakasunod-sunod daw ng mga bases na yan, yung A, C, C, A, depende. It's a very hindi na siya very random. I mean the exact sequence of these nitrogenous bases can define a protein, an amino acid actually. Anumang pagbabago sa arrangement ng mga nitrogenous bases na yan will cause changes sa mapoproduce nating amino acid. And once we change this amino acid, we will also be changing the final protein na mapoproduce natin. At once na nagbago yung protein, may mutation na naganap, pwede siya makaapekto sa health natin. And that's the reason why may mga genetic diseases tayo na like yung tinatawag nating G6PD, na natetest sa newborn screening, if you're familiar with that one, yung sakit na yan na nahihirapan yung katawan nating i-metabolize yung sugar, ang nangyari lang doon is there's some changes here sa sequence ng ating nitrogenous bases. Imagine that one. Isang maliit na pagbabago lamang ang nangyari sa gene na nagme-metabolize ng sugar na yon, and at the end of the day, ang laki ng effect na niya sa whole organism. So napaka-specific, napaka-importante, napaka-essential nitong sinasabi nating linear order of sequences ng ating mga bases or ng ating mga nitrogenous bases. This diagram will be the main topic next week. Yan, itong simpleng diagram na to, ito ang magiging focus natin next week. Okay? Aside from that one, banggit na rin natin kahapon, and I think nabigyan naman natin ng hostisya sa discussion natin, that based from the different experiments and researches conducted by several scientists starting from Rosalind Franklin up to the two guys, James Watson and Francis Crick, we now know that our DNA is composed of 2 strands, 2 anti-parallel strands running and forming a double helix. To give you an idea again, pag sinabi nating double helix, it's like a ladder na tinwist natin, inikid natin. So ganyan ang itsura ng double helix structure ng DNA. Pero sa performance task ninyo, na I think na I just hope na nabuksan nyo na yung performance task nyo, do not expect that you're going to extract DNA sa halaman na ganyan ang itsura. Huwag kayong mag-expect na makakita kayo ng twisted ladder formation or structure sa DNA na i-extract ninyo sa inyong experiment/performance task. This one is an ultrastructure. Pinapakita nito yung pinakamaliit or pinakapinong structure ng ating DNA. I will not go over the details yung i-expect nating ma-extract sa mga halaman o sa mga sources natin para at least ma-enjoy nyo yung experiment na ipapagawa ko sa inyong performance task. By the way, yung performance task natin magkakaroon ng changes sa groupings dahil nga po may mga napili sa face-to-face. So, I will just update the file ng ating groupings. Now, the 2 sugar phosphate backbones are on the outside of the helix. Kaya nga backbone diba? Nasa labas siya. Just like our backbone sa likod ng ating katawan. And the nitrogenous bases are paired in the interior of the helix. Going back to this diagram, kita nyo po, this blue strands or this blue ribbon is the sugar phosphate backbone. And inside our sugar phosphate backbone, you can see here the bases na magkakapares. At hindi sila randomly magkakapares may specific pairings. For example, the 2 strands are held together by hydrogen bonds between the paired bases. Thymine is always be paired with adenine using 2 hydrogen bonds. If you still remember, pag sinabi nating hydrogen bonds, these are kinds of bonds between a very attractive element, just like oxygen, to hydrogen. In this case, Ayan po, dalawa po ang strand o dalawang hydrogen bond ang nabuo. And also, cytosine will always be pairing to cytosine using 3 hydrogen bonds. Kaya mapapansin niyo sa ating drawing, ay hindi pala visible sa diagram to. Nevertheless, here, in this diagram, you can see here we have 3 hydrogen bonds between cytosine and guanine. This is a major concept sa biotechnology na sana maalala niyo po. Thymine will always be paired to adenine, or T is paired to adenine, and cytosine will always be paired to guanine. Again, T to A, then C to J, C to G. Hindi sila randomly magpapares si thymine, magpapares kay guanine, si A kay C. No. Magkakaroon ng mutation pag ganun, or there will be some errors. There will be major errors sa ating sequence. So laging T paired to A and C paired to G. Okay, so in the base pairing, only certain bases in the double helix, ayun nga sinasabi ko kanina, are compatible with each other. Adenine in one strand always pair with thymine, just like with that diagram. And guanine always pair with cytosine. Laging ganyan po ang pairing. Yes, there's a tendency na magkaroon ng mga mispairings o magkakamaling pairings that usually happens due to radiation, due to some mutations, so and so forth. Pero in natural, in normal setting or normal condition, A will be paired to T and C will be paired to G. Maliwanag tayo dapat dyan. So let's review. Adenine and guanine are type of nitrogenous bases called purines, pyrimidines, or nucleotides. Again, adenine and guanine are type of nitrogenous bases called purine, pyrimidine, nucleotides. What do you think is the answer, or what do you think is the missing word in our statement? Please use the comment section for your answer. I will wait for your answer right now. What do you think is the term that will complete our sentence here? Adenine and guanine are type of nitrogenous bases called blank. Jane answered pyrimidine. What about the other students? What do you think is the answer? We have 3 choices here. Marcus answered purine. Yan, most of you are answering currently now purines. Rian, Kisha, Micah, Joshelle, and or Jamil are answering purines. Okay lang po magkamali. Do not delete your answer. It's okay to have mistakes here. Pare-paro tayo natututo. Okay? So yan, the answer for this one is purines. Again, the code is PAG and PICAT. Yan po yung mnemonics natin para maalala yung pinagkaiba ng purines and pyrimidines. Next, deoxyribose, unlike ribose sugar, lacks a blank group in one of its carbon. Is it carbonyl, carhydroxyl, or carboxyl? Again, the statement is deoxyribose, unlike ribose sugar, lacks a blank group in one of its carbon. Is it carbonyl, hydroxyl, or carboxyl? What do you think is the answer? Richard answered hydroxyl with question mark. Yan. Jane answered hydroxyl. Okay, let's see what is the answer for your other classmate. Joshelle answered hydroxyl. Marcos hydroxyl. Rian, Jamil answered hydroxyl. The answer is hydroxyl. Walang hydroxyl group. Tanging hydrogen lamang po ang nasa carbon number 2 ng deoxyribose natin. Next, let's have another one. Adenine in one strand always pair with blank. Uracil, thymine, guanine. Again, adenine in one strand always pair with blank. Uracil, thymine, guanine. What do you think is the pair? O sino nga ba ang laging kapartner ni adenine? Yan na, basic dapat na ito sa inyo. Kung sino ang kapartner lagi ng mga bases natin. The answer for this one is thymine. A to T, then G to C. Next, the 2-sugar phosphate is arranged in a blank orientation in which the backbone run in opposite direction from each other. Is it semi-conservative, parallel or anti-parallel orientation. Again, the statement is the 2-sugar phosphate is arranged in an orientation in which the backbone runs in opposite 5' to 3' direction from each other. Is it semi-conservative, parallel, or anti-parallel? Yan, Levi, Richard, Marcus answered anti-parallel, as well as Jomel, Micah, Colleen. Hindi ko na isa-isa, marami na sila. The answer is, I think most of you are answering anti-parallel right now. The answer is anti-parallel. Magka-opposite kasi sila. One is running 5', both of them will be running 5' to 3', pero magka-opposite yung direction ng strands nila. Kaya siya tinawag na anti-parallel. Again, the significance or the implication of this orientation will be further discussed next week sa lesson number 2. Yan. So the last part here, the 2 strands of the double helix are complementary to each other. Pag sinabi natin complementary, ano kaya ibig sabihin nun? Grade 8, let me ask you, vocabulary building tayo ngayon. Ano yung word na naiisip ninyo kapag naririnig nyo ang word na complementary? What are the words that are synonymous to the word complementary? Synonyms lang tayo. Ano yung mga words na naisip nyo pag naririnig nyo ang salitang complementary? Or complement. Hindi complement ha, complement. Yan, Richard answers specifically for that thing or object. Wow, napaka-sweet naman. Specifically for that thing or object. What about the other students? Anong naisip nyo pag narinig nyo ang complementary? Combining, sige. Okay. Let's see. Compatible, yes, sige. Compatible daw. Yan, that mahasa yan. Mahalaga na sa science subjects natin na hasa ang English or vocabulary skills natin. Mahalaga po yan. It's a skill. Okay? So, let's use the term na ginamit ni Richard. Sabi ni Richard dito, specifically for that thing or object. Simply compatible sila, di ba? Made for each other. Parang ganun lagi ang DNA strands natin. Sabi dito, the 2 strands of the double helix are always complementary to each other. Meaning, kapag meron tayong isang strand ng DNA, malalaman na natin kagad ang kapartner niya. Bakit? Kasi using this one, for example, isang strand lang yung binigay, this one, ito lang yung binigay sa inyo. Alam natin ang kapartner ng A is T. Ganyan, di ba? Ang kapartner lagi ng T is A. Kapartner ng G is always C. And kapartner ng C is G. Ibig sabihin, very specific. Kung sino yung nasa unang strand natin, malalaman, matutukoy kagad natin yung kapartner na strand kagad niya because they are complementary to each other. They are compatible, or as to be specific, lagi silang magkapares. Aside from that one, as you can see here, one strand is running 5' to 3' and the other one is 5' to 3' because of its anti-parallel orientation. Again, A will always be paired to T and G will always be paired to C. So, let's have a review. Tingnan natin. If one strand of the DNA is 3' AGT 5', its complementary strand must be A, B, or C. Isipin natin ha, just magbigay kayo ng oras, isipin ninyo, ano kaya ang kapartner na strand ng 3' AGT 5'? Is it A, B, or C? Again, use the comment section for your answer. What do you think is the answer for this one? Jane answered B. What about the other students? Is it A, B, or C? Again, the strand is 3' AGT 5'. Yan, dapat ma-master nyo to ha. Kasi next week, puro ganyan halos na pag-aaralan natin. Kasi puro letters na ganyan. So yan, tingnan natin. The answer is letter B. 5' TCA 3'. Bakit? Bakit sir? Check natin ha. 3' to. So 5'. Check. The last strand here is 5'. 3'. Check na yan. Sabi dito, it's A. Ang partner doon ng A is T. Tama. G, C, T, A. Yeah, the answer is letter B. Let's have the next one. If one strand of the DNA is 5', CCG3', its complementary strand must be, ano kaya ang sagot dito? 5' CCG3'. The answer is it A, B, or C? What do you think is the answer for this one? Isipin muna natin, you can use a paper if you have 5' CCG 3'. What do you think is the complementary strand nitong isang to? Make sure na anti-parallel sila ha, and also the correct pairing. Let's wait for the answer of your classmates. Ano kaya ang sagot nila sa item na ito? Ayan, na-delay pala yung comment section. So all of you are currently answering A. The answer is A. 3', 5', then G, G, C. Madali lang naman yan eh. Pero next week, ako, good luck talaga. Mahirap yon. If one strand of the DNA is 5' TCA 3', its complementary strand must be blank. Again, the strand is 5' TCA 3'. What do you think is the answer? What is the complementary strand of this DNA? Ano kaya ang sagot dito? Lahat ng items natin dito, it's 3' to 5'. So, ano kaya dito? Alin kaya dito ang tatama? Okay, Jane, Joshelle, Marcus, Rian, hindi ko na isa-isa yung mga names. All of you are answering currently right now C. What do you think is the answer? The answer is letter C. 3' to 5', then A, G, T. Basic pa lang yan, tatlo-tatlo lang yan. Sa inyong activity yata, 6 or 9 yata yung strands natin or some yung bases. I think you can do that one. So yan. So I just hope that you have now a good grasp of the concept regarding the structure of our nucleic acid. So, ang tanong dito, alam natin na yung DNA na yan ay genetic material ng isang cell. And we all know that the genetic material, specifically the DNA, is a very important molecule, kaya siya nilalagay sa nucleus. But the problem here is that our DNA, kapag in-stretch natin ang isang DNA na nasa loob ng nucleus ng isang cell, it will take, sa tao lang ha, it's 5 meters in length. Ganun kahaba yung DNA ng isang tao. So ang tanong dyan, grade 8, paano kaya siya nagkasya sa loob ng nucleus natin gayong napakaliit ng cells ng ating katawan and the length of the DNA is 5 feet ang haba? Let me ask you sa comment section, what do you think is the idea? Paano kaya napagkasya ng ating cell yung DNA sa loob ng mas maliit pang nucleus? Let me ask you, anyone who has an idea, paano kaya napagkasya natin ang yung DNA na yan sa loob ng ating nucleus ng ating cell, which is a very long molecule, 5 meters. Pero manipis yan ha. Baka sabihin nyo napakalaki niyan. It's a 5 meter length, 5 feet pala, sorry. 5 feet ang haba ng ating DNA. Paano siya napagkasya? Looping way or circular folding, aba? Mga may nagsasearch sa internet, mga sa internet sinestra. Idea nyo lang, I mean, your first guess. Kasi napakahaba ng DNA natin, paano siya mapagkakasya? But anyways, Richard's answer is correct. Pag-loop at pag-fold. Maglo-loop yan, mag-iikot-ikot yan. Yan ang pag-aaralan natin at this moment sa last part ng discussion natin for this afternoon. So here, the main component of the genome in most bacteria is one double-stranded circular DNA molecule that is associated with a small amount of protein that causes the chromosome to coil and supercoil, densely packing it so that it fills only the part of the cell. Tapos na, ito na yung mismong summary ng discussion natin sa part na ito ng ating lesson. Okay? Sabi daw dito, yung DNA ng isang typical bacteria, circular. Alam na natin yan, didiscuss na natin yan ng first at second grading period. Pero, yung DNA natin, it is not just pure DNA. It also contain proteins, mga protina na nakakatulong para, hindi na siya buhol-buhulin, paikot-ikutin sila hanggang sa ay umikli ng umikli ng umikli hanggang sila ay magkasya sa loob ng ating cell. Sa case natin sa eukaryotes, para siya ay magkasya sa ating nucleus. So napaka-special ng relasyon ng DNA at protein sa case na ito. Kasi without this protein, our DNA will be very long, very lengthy material at baka hindi ito magkasya sa ating cell. So each eukaryotic chromosome in a sense naman contains a single linear DNA double helix that in humans averages about 1.5 times 10 raised to 8 nucleotide pairs. So around 1.5 billion pairs. Imagine the number of those A, T's, and G's and C's sa ating nucleic acid. Ganyan karami. So napakahaba talaga ng DNA natin. This is an enormous amount of DNA relative to a chromosome's condensed length. So titingnan natin yung mga concepts na binabanggit ko po ngayon ito. So paano nagkasya ngayon talaga itong DNA na ito sa ating napakaliit na nucleus ng isang cell? So in a cell, eukaryotic DNA is precisely combined with a large amount of protein. So hindi natin masisisi yung mga unang scientists natin kahapon, sina Frederick Miescher, sina Thomas Hunt Morgan, kung bakit every time na nag-extract sila ng DNA, laging may kasamang proteins. Because integral ang connection or very essential yung connection between your proteins and DNA in a sense sa isa ating cell. Together, this complex of DNA and protein is called chromatin. So yan ha. So DNA helix, kapag sinamahan na natin na siya ng mga proteins, it will now be called chromatin. These long thin strands of chromatin, so mahabang strands yan, will start to condense forming a chromosome. Ayan, very basic yung concept niya. We have a linear strand of DNA. Kapag sinamahan natin siya ng proteins, it will be chromatin. It's a very long, thin strands. At once na nagkumpul-kumpul na sila, nag-start na sila mag-coil, mag-loop, and mag-start na sila mag-fold, they will now form chromosomes. DNA, chromatin, chromosomes. So, chromosomes are discovered in 1843 by Carl Wilhelm von Nageli, by using special stain that makes them appear to have colored bands. Colored bands yan ha, hindi colored hands, sorry. Kaya siya na tinawag na chromosome. Chromo means colored, so means bodies, colored bodies. So during his time, gumamit siya ng mga special chemicals na nagbibigay ng bands, iba't ibang kulay sa mga genes na yan na nasa ating chromosomes. Kaya siya tinawag na chromosomes. These colored bands of the chromosomes are the specific location or locus of different genes. So ibig sabihin isang band, di ba napansin niyo itong dark light bands na yan. Ang isang band na yan, for example, this is your chromosome, ito yung dark band na yan, that is a gene. So gene 1, gene 1. Itong light band, that is gene 2. So ibig sabihin lahat ng yan, yung mga yan, yung mga colorations na yan, yung mga bandings na yan, yung mga locus na yan, is a specific location for a gene, for a specific gene. So that should be clear na sa inyo yan, yung part na yan, okay? Using special stains, actually kaya natin sanang gawin yan sa laboratory. I have my chemicals with me. Pwede tayong i-extract natin yung chromosomes na yan sa fruit fly at pwede nating gawin yan. Kaya lang kasi, yun nga, nasa online tayo pare-pareho, so we cannot do that one anymore. Pero may mga simulations. I will look for some simulations wherein you can do these experiments using computer or your gadgets. Just remind me later. Right? So, ngayon, going back to my first question kanina, paano na ipapasok yung napakahabang DNA na yan sa loob ng ating nucleus? The answer here is what we call a multi-level system of packing. So, ibig sabihin, parang ganyan, di ba, pag nag-i-impake tayo ng gamit sa maleta, napakadami nating inilalagay sa malete at the end of the day, hindi nagkakasya. So ang gagawin nyo ulit, tatanggalin nyo ulit yung mga damit, yung mga nilalagay nyo sa maleta at iniayos nyo. Iniisa-isa nyo i-impake ng maayos, mas na ipapasok natin ng mabuti yung mga damit na yan sa ating maleta. Pero pag yan very random, pinasok lang natin ang pinasok yung mga damit na yan sa maleta, most of the time or actually at the end, hindi magkakasya yung material na yan, yung mga damit na yan. Pero kung systematic nating ipo-fold, systematic nating itutupi, systematic nating ipapasok sa maleta yung mga DNA na yan, magugulat kayo na yung same amount ng material na yan ay magkakasya sa maliit na lalagyan. Yan din po yung concept sa tinatawag nating multi-level system of packing. Okay? So, nabanggit na natin kanina that there are some proteins that are very important in this multi-level system of packing. And these proteins are called histones. Proteins called histones are responsible for the first level of DNA packing in chromatin. So yung mga histones na yan, actually 8 piraso yan, may mga names yan, we will not just go over the names, just remember that there are histones. They will be, sila yung parang iikutan ng ating DNA. Very obvious naman sa ating diagram. Siyempre, pag inikot natin yung ating DNA sa mga histones na to, i-click agad yung DNA natin. Diba? Kasi inikot na natin ilang ikot yan. I think it's 1.5, kung hindi ako nagkakamali, 1.5 times na iikot siya sa mga histones na yan. So, i-click agad yung DNA natin once na umikot siya or nag-coil siya sa ating mga histones. H2A, H2B, H3, and H4 are the names or these 4 main types of histones, and these 4 types of histones are critical to the next level of DNA packing. So once na nagsama na po yung DNA at histones, ang tawag po dun ay nucleosomes. Going back here to this diagram, ito po yun. This circle is yung histones natin, and the DNA is here, nakaikot na dyan. So again, pag sinabi nating nucleosome, it's DNA plus histones. DNA plus histones. Yan. So, yan. Clear na tayo dyan, ha? Ngayon, siyempre, pag inikot natin yan, pwedeng matanggal yung pagkakabuhol o pagkakaikot ng ating histones. So, kailangan natin ng another set of molecules para siya ay mapagdugtong natin. So, yan po yung H1 histones natin. Ito po yun. Para siyang yung pang-secure, para siyang lock, para hindi matanggal yung pagkakabuhol ng ating DNA sa histones natin. Nucleosome is the basic unit of DNA packing. The string between beads is called a linker DNA. So, in this case, this is your linker DNAs, and this is your histones, itong pink ones, and this is your another H1 histone. Ito pa yung lock para siyang nagsisecure, para hindi matanggal yung pagkakaikot ng ating DNA sa ating mga histones. Siyempre, kailangan pa natin siyang paikliin. The next level of packing results from the interaction between the histone tails of one nucleosome and the linker DNA and nucleosome on the other side. So, magkakaroon ng interaction, yung magkakasunod ng mga nucleosome, at iikli lalo sila, forming what we call a 30 nanometer fiber. 30 nanometer fiber kasi yung lapad niya will become 30 nanometer. Yun nga lang, mapapansin niyo, every time na umiikli yung DNA natin, mas tumataba siya ng kaunti. Siyempre, nabubuhol siya ng nabubuhol. You can try this one out. Kuha kayo ng lubid, ibuhol yung one end, mapapansin niyo mag-i-start na siya mag-coil ng sarili niya. Pwede niyo siyang itry. I-try niyo yung cord, start coiling, then after a while, mapapansin niyo habang humihigpit na siya ng humihigpit, nag-i-start na rin umikli yung ating lubid. Ganyan po yung nangyayari sa ating DNA. With the help of these proteins, these histones, linker proteins, mga ganyan, the DNA will start to shorten and it will become thicker. May possibility po ba na mag-snap? Definitely. Definitely, iho. Pwede siya mag-snap, yung mga nucleosomes na yan. Kaya nga, very careful ang packing na ginagawa. Napakababusisi ng pag-unpack at pagpapack ng ating DNA. Pwede siya mag-snap, definitely. Lalo kung may mga problems yung sa DNA natin, sa mga histones natin, pwede po. At lalo sa mga loose ends ng DNA natin, sa mga dulong parte, pwede talaga siya magputol-putol. The 30 nanometer fiber in turn forms loops called loop domains. So magsisimula sila mag-loop. Diba, nagbubuhol-buhol na sila, then mag-i-start sila mag-loop. So iikli na naman yung DNA natin, pero tataba siya. Yun ang rule, ha? Kanina, mahabang-mahaba pero manipis. Nung nagkaroon ng nucleosome, umikli siya pero medyo tumaba siya. Ngayon, 300 nanometers na yung lapad ng ating forming chromosomes. And this will form a 300 nanometer fiber. So tuloy-tuloy lang po yan. Magbubuhol yan ng magbubuhol. Napansin may mga protein involved pa rin dyan. Not necessarily histones. Mayroon na siya mga protein filaments that will help in forming these loops or these loop domains. Then eventually, we are now going to have now the chromosome. Tuloy-tuloy lang po yan. Mag-loop, mag-fold, magkakaroon ng mga linkages between the proteins until such time that they will form a chromosome. Very visible ang ating chromosome sa metaphase stage. If you still remember yung metaphase stage ng ating mitosis, yung ating ang chromosomes ay nasa gitna ng ating metaphase plate. I just hope that you still remember that topic. If you can't remember, go back to that video lesson. In a mitotic chromosome, the loop domains themselves coil and fold further, compacting all the chromatin to produce the characteristic metaphase chromosome. Yun yung parang butterfly shape na very familiar. Pag nag-isip kayo ng chromosome, yung kagad ang idodrawing nyo, parang butterfly na yan. That is your metaphase chromosome. And during metaphase, magiging super condensed yung structure na yan at mas magiging visible na yan sa mga cells natin. At yan yung pinag-aaralan ng ating mga geneticists at cytologists para ma-detect kung may mga genetic abnormalities yung mga bata. O lalo yung mga tao. Yung mga Down syndrome, yung trisomy 21, yung mga iba't ibang uri ng mga genetic disorders or genetic abnormalities can be determined using this technique or using this chromosome, the metaphase chromosome. So typically, your metaphase chromosome is composed of two arms, the long arm and the short arm, or simply the P arm and the Q arm. I just forget kung bakit nga pinangalanan siyang P or Q arm, or I will just try to look for my notes regarding this one. Pero in the sense, it has a short arm or the P arm and the Q arm. And at the middle, we have the centromere. The centromere is actually a proteinaceous structure kung saan nagdudugtong yung dalawang pares ng DNA natin. Kasi tandaan nyo, replicated yung mga DNA na yan. Doble. Okay? P arm and Q arm. So during metaphase, okay, all the DNA condenses into 23 pairs of chromosome. 22 will be considered as somatic chromosome, while the remaining one pair is considered as sex chromosome. And that one pair will determine if you're a female or a male. Kapag ang nakuhang chromosome natin is XX, diba? Yung sperm ni tatay, ang daladala niya is X. Ang egg ni nanay, ang daladala is X. X plus X, you're going to get XX. Pag yan nakuha nyo, female. Kapag naman yung sperm ng tatay, ang daladala niya is Y, ang nanay naman is always X naman. Pag nabuo natin yan is XY, ang mabubuo naman natin dyan is lalaki, male. So in this case, this set of chromosomes, ang tawag dito, itong arrangement na to, ang tawag dito ay karyotype. Sayang, pinapagawa ko to sa mga estudyante ko sa face-to-face. Pero alam ko may mga online activities wherein you can do this one. This one, babae o lalaki kaya ito? Let me ask you sa comment section. This karyotype showing the 23 pairs of chromosome, is it a male or a female individual? Let's have the comment section. Let's see. Babae o lalaki kaya ang taong ito or individual na ito? Let's see sa comment section. What do you think is the gender or the sex of this individual? Is it a male or a female? Check nyo yung ito yung sex chromosome natin nasa dulo para magkaroon kayo ng idea. Is it XX or XY? Richard answered male. Joshelle answered female. Dalawa lang ng choices natin. So sino kaya ang tatama sa kanilang dalawa? Anyone? Sino pa ang gusto sumagot? Is it a male or a female? Yan, may mga utang tuloy akong mga links mamaya. I-share ko mamaya yung links para sa ating inyong mga online activities, yung mga ganitong simulation. Levi answered male. The answer for this one is female. Kasi dalawa yung X niya. X, X. Pero kung ang nandito yung isang Y, yung nandito yung isang chromosome, wala ito, yan, wala yan, it's XY, lalaki yan. So the answer for this one, it's a female. Dalawa kasi yung X chromosome niya. Pero pag yung isa ay X, isa ay Y, that's a female, that's a male one. Just like this one. Okay? Again, XX is female, XY is male. So paano natin nabuo yan? Paano natin nagawa itong series of arrangement of chromosomes na ito at metaphase? The process is what we call karyotyping. Karyotyping is the process of pairing and ordering all the chromosomes of an organism. So in a case sa atin, 23 pairs ang i-arrange natin. So anong gagawin ng mga scientists dyan? Ang gagawin nila dito, ayan yung several process niyan. We will get blood samples. They will stimulate mitosis using chemicals, and they will stop mitosis at metaphase. Bakit ini-stop natin sa metaphase? Dahil very condensed yung ating mga chromosomes during metaphase. Mas visible sila. Then they will culture it, they will put colors, stains, and all those things, at ilalagay siya sa microscope at iisa-isahin siyang i-arrange under the microscope. Some cases, digitize na yung pag-arrange na yan to form the karyotype. And after that one, dyan natin malalaman kung ano ang gender at ano yung sakit ng individual na tinetest natin. So ang tawag dito is karyotype, and the process is called karyotyping. Karyotypes are prepared using standardized staining procedures that reveal characteristic structural features for each chromosome. May mga specialized tayong mga hospital. I think St. Luke's, I think that's a specialized hospital that can do this karyotyping. Pero marami na ring hospital na gumagawa ng karyotyping to determine if that individual, if that infant or baby has a genetic disorder. I-arrange lang nila yan. As in literal na pagpaparis-parisin nila, then they will check if there's abnormalities. Kasi for example, sa trisomy 21 or Down syndrome, Down syndrome tayo ha, sa Down syndrome, sa trisomy 21, tatlo yung ganito nila. Tri means three, somy means body. So tatlo yung trisomy 21 niya. So that's a problem. So this one, for example, meron siya dito, we can say na this individual is a female with Down syndrome. Meron naman doble yung 5, nawawala yung isang pares, ganyan. So please, I'm asking you to watch the mutation videos sa Prism YouTube channel because that will explain further this topic. Can karyotyping sometimes? Oh, definitely. Pwede naman siya magkamali. Kaya nagkapa-second opinion tayo. So obviously, for example, nalaman na may genetic abnormalities na triple yung isang chromosome natin, gusto siyempre natin magpa-second opinion. You will go to another facility. But I think it's very I'm not sure about the price or the cost, but I think it's a very pricey process. Medyo mahal. Kasi siyempre, isa-isa i-analyze, i-confirm pa nila yan. And I think it's also prone to some mistakes. Pero siguro iniiwasan rin nila yan kasi nakasalalay dito yung health ng baby or ng individual na tinetest natin. So pwedeng mag-doble itong meron pa nga tayong sakit, poly-XY, poly-X female, 4 yung X chromosome niya. Meron tayong poly-X, ano ba yung mga sakit pa? Nawawala yung isang chromosome, doble-doble yung chromosome, triple. Ito poly-XY, sobra-sobra. Meron siyang walang Y. Isa lang yung X chromosome niya, X0. Turner syndrome yata ito. So marami siyang sakit, maraming genetic abnormalities na nakakonekta dito sa chromosomes na ito. Okay? So you can discover that one by watching the YouTube video ko po regarding mutation. Let's move on. So chromosomes can be classified into 4 main groups based on the location of the centromere. Kapag yung centromere niya, check lang natin Kapag yung centromere niya ay nasa perfectly gitna, pantay yung haba ng P at Q arm, metacentric ang tawag. Metacentric. Meta means middle. Okay? Kapag naman yung yung centromere natin medyo malapit sa taas, so mangyari sobrang ikli itong P arm at sobrang haba ng long arm, ang tawag naman dito ay submetacentric. Sa acrocentric naman, meron siyang maliit na parang stub dito sa taas. Meron pa siyang counting P arm. That one is called acrocentric. At naman pag totally nawawala na yung ating upper arm or short arm, at ang natitira na lang is yung centromere and the lower arm or the longer arm, ang tawag naman dun ay telocentric. I think sa activity ninyo, ika-classify nyo siya kung ito ba ay meta, submeta, acro, or telocentric chromosomes. Again, after this one, please watch the mutation YouTube video natin sa Prism para kasi yun yung activity number 4 natin. So let's review. I think this is the last part of our discussion for this afternoon. Let's review. Bacterial chromosome is blank, unlike eukaryotic chromosome, which are linear. Is it parallel, circular, or helical? Again, bacterial chromosome is blank, unlike eukaryotic chromosomes, which are linear. Is it parallel, circular, or helical? What do you think is the answer for this statement, or what do you think is the missing word from this statement? Richard answered circular with question mark. What do you think is the answer? Keisha answered circular. Rian circular. I think siguro majority will be answering circular. So let's see if the answer is circular. Pareho silang helical. Bacterial chromosome and eukaryotic chromosome are both helical, but eukaryotic is linear, bacteria is circular DNA. Next statement. Proteins called blank are responsible for the first level of DNA packing in chromatin. Is it histamine, linker protein, or histones? Again, the statement, proteins called blank are responsible for the first level of DNA packing in chromatin. Is it histamine, linker protein, or histones? Let me ask you, what do you think is the missing word? Joshelle answered histones. Yan, Levi, Rian also answered histones. Ayan, Marcus also answered histones. The answer is histones. Yung linker proteins yung nagdudugtong kanina, that's also histones. Pero siya yung nagdudugtong. Histamine is an allergy molecule. Siya yung nagkukos ng allergy. Next, blank is the process of pairing and ordering all the chromosomes of an organism. Again, the statement is, blank is the process of pairing and ordering all the chromosomes of an organism, cross-matching, karyotyping, or staining. What do you think is the answer for this one? Richard answered karyotyping. I just hope na nandito pa kayo, kasama ko pa kayo sa ating online class this afternoon. Jane, Levy, Colleen, ayan, karyotyping po ang sagot dyan. It's a very laborious process wherein we're going to pair and order all the chromosomes of an organism. Next, during metaphase, sa tao ito sa tao, all the DNA condenses into blank somatic chromosome and one pair of sex chromosome. Is it 22 pairs, 23 pairs, or 46 pairs? Again, during a metaphase, all the DNA condenses into blank, which is a blank somatic chromosome and one pair of sex chromosome. Richard answered 23. Some, but the rest of you are answering 22 pairs. So sino kaya ang tama sa inyo? The answer is 22 pairs. Paano sir? Linawin nga natin ulit ito. This is 22 pairs plus another pair, kaya siya nagiging 23 pairs. So all in all, ang number ng chromosome ng tao is 46. That is the 2n number of chromosome natin. Okay, maliwanag na dapat sa atin yan. Next, metaphase chromosomes are basically composed of arms attached to a blank, kinetochore, centrosome, or centromere. Metaphase chromosomes are basically composed of arms attached to a blank, kinetochore, centrosome, or centromere. Richard answers centromere. Again, after this one, please, if you want, rewatch the videos bago kayo magsagot. Huwag tayong ma-excite sa pagsasagot ng mga activities natin. Yung iba dito, akala nila naalala nila lahat. They will go to the activity sheets and everything will be wrong. The answer is centromere. So this ends our discussion. Again, the soft, bago tayo umalis bago tayo bumalik sa main platform, reminders lang po. Sa Friday po, soft deadline. I will not check the link. Hayaan ko lang kayo magsagot. Pero gawin niyo po siyang personal deadline para hindi kayo matambakan ng trabaho. Within this week, kailangan masagutan na natin at least ang pre-test, lesson 1 activity, and quiz number 1. But the hard deadline of all the links and performance tasks will be April 18, 2022 pa, Monday, 12 noon. Pero wag niyo na pong hintayin yung April 18 para sagutan yung mga activities na yan. Okay? Every week, sagutan niyo pa unti-unti kasi after the 5 lessons, performance task naman ang tatrabahuhin ninyo. Okay? So I hope everything is clear. I will go back to the platform na. Just give me 1 minute. I will give you some reminders regarding our lesson. Ayan. So, thank you for attending our lesson for this day. Again, reminders lang po. Mamaya mag-share po ng mga videos or simulations pala rather. Simulations wherein you can try DNA extraction and this karyotyping. I just hope na makakita ko ng magandang simulation. Then also, after this one, paanoorin niyo po yung video natin about mutation. I think nakasend na yung link na yon sa ating PDF file. Okay, so panoorin niyo po yon para masagutan niyo rin po yung complete set ng activities para sa lesson number 1. Aside from that one, within this week or after this week, kailangan masagutan na po natin ang pre-test, lesson 1 activities, and lesson 1 quiz number 1. Okay? So again, nagpapasalamat po sa pag-attend niyo po ng ating lesson sa hapon na ito. I hope to see you next week. I hope na mas marami tayo next week. Kasi ako na po nagsasabi, mas mabusisi ang lesson natin next week. Okay? So, i-update ko kayo sa mga magpe-face-to-face kung mag-i-start na tayo sa lesson 2 next week. Kasi na-discuss ko na yung lesson 1. I will not repeat this lesson 1 next week. So most probably, ang start na po ng lesson natin is lesson number 2, which is the central dogma of molecular biology. So I hope na panoorin nyo muna ito, baka wala akong kausapin next week sa face-to-face. Kung kailan tayo mag-i-start ng face-to-face natin. So again, maraming salamat sa pagdalo ng ating online class and I hope to see you more next week. Again, goodbye.